In class, we looked at how prokaryotes reproduce and how they maintain genetic variation. In this video, we're going to take a look at how viruses replicate and how they maintain genetic variation. Let's quickly review viral structure. You don't have to draw this diagram in your notes because you've probably already got it there somewhere. Remember that viruses are very simple. They have DNA or RNA inside, and then they have a capsid or a protein coat. And oftentimes, if you're a bacteriophage, that might be divided into a head and a tail. And all viruses have receptor proteins that they use to attach to their host cell. So one way that viruses replicate is through the lytic cycle. Let's start at the beginning. Here we have a bacteriophage, a virus that infects a bacteria, and the first thing it has to do is attach to the bacteria cell using its receptors and then inject its DNA into the cell. Once the DNA or the RNA is in the cell, it can take command of the host cell, and now it's going to use the host materials, like its enzymes and other proteins, to make more viruses. So here you can see the DNA from the virus has been used to make more viruses. And once all those viruses are complete, they are released from the cell through a process called lysis. The viruses burst out of the cell, destroying the host cell in the process. The other way that viruses replicate is the lysogenic cycle. And here it starts the same way. We have the virus attaching to the host and injecting its DNA. But here something different happens. The virus is going to insert its DNA into the host cell's DNA. And at this point it's called a prophage or a provirus. And it might just hang out there for a very long time. And as the host cell reproduces, it's going to reproduce the viral DNA as well. And after a while, you might have a lot of host cells all containing viral DNA. And at any point, this virus could switch back into the lytic cycle, start taking over the cell, and reproducing more viruses. Now let's take a close look at HIV, because HIV works a little bit differently. If you look at its structure, first of all, it's got an extra layer around its protein coat. It's got an envelope with glycoproteins. Those are sugar proteins. The other interesting thing about HIV is that it has RNA as its genetic material, not DNA. It also has a special enzyme called reverse transcriptase, which does just that. It transcribes RNA back into DNA. So because viruses have RNA as their genetic material, they're known as retroviruses. And let's see how a retrovirus like HIV replicates. So we have viral RNA, which is in the cell. First, reverse transcriptase uses the viral RNA to make DNA. Then it makes a second DNA strand. Now this DNA strand is going to insert itself into the host cell's DNA, becoming a provirus. And it may hang out here for days, months, even years, which is why some people with HIV don't actually show AIDS right away. But then a trigger might cause the virus to enter the lytic phase. And so the viral DNA will be transcribed into RNA, and then the RNA will be translated to make viral proteins. And then finally, the virus will lyse and leave the cell. So here we see that HIV follows both the lysogenic piece and it can follow the lytic replication cycle. So finally, how do viruses achieve genetic variation? Well, first thing is that mutations that occur at a higher rate in viruses because they don't have proofreading enzymes. So during DNA replication and transcription, errors that occur are not caught and are not fixed. The other important thing is that if two viruses infect the same cell, the viral DNA might recombine into new viruses. And so because of this, bottom line, viruses can evolve very quickly in response to selecting agents.